In our first chapter here on land plants, we're going to talk about how plants colonize land. It's just within the last 500 million years or so that animals, fungi, and land plants did colonize dry land. So if we look at where plants came from, our biggest clues come from algae that are known as charophytes. And charophytes are the closest known relatives of land plants. So these are the ones that they seem to share that common ancestor with most recently. So when we look at charophytes, we can get some clues as to what adaptations might have been necessary for plants to make the move onto dry land. Charophytes themselves share a number of unique characteristics with land plants. So if we take a look at these unique characteristics, these, along with some molecular data, do support that idea that these are the closest relatives of land plants. So together, they both have rings of cellulose synthesizing proteins. This is going to be something that's in the plasma membrane. And this is different from other algae because other algae, they will have cellulose synthesizing proteins, but they're going to be linear proteins. So this ring thing is unique to the charophytes and then also the land plants. They also are both going to have peroxisome enzymes. This can be very helpful when we're dealing with the effects of photorespiration. Another thing is that they have a similar structure to their flagellated sperm. The last one that they share in common is the formation of a phragmoplast. This phragmoplast is a collection of microtubules, and this forms between daughter cells of mitosis. So this is something that's part of the cytokinesis process. This is going to give rise to that cell plate that will later develop into the cell wall that will separate the two daughter cells from each other. So these four characteristics are unique to land plants and charophytes, and this supports that close relationship between the two, and it's also supported by some molecular data. In this picture, we're seeing charophytes right underneath the water here. So those charophytes are in shallow water, and because it's shallow water, sometimes the water will recede and it will leave them dry, and they'll have to be able to deal with that temporary dry period. So charophytes did have a few unique adaptations that enabled them to withstand periods like that. So one of those was the development of what we call sporopollenin. Sporopollenin is a polymer that is very durable, and it covers the exposed zygotes of the plant, and it helps them to not dry out or not get dehydrated during those periods of dryness. There is sporopollenin that's found surrounding spores of plants today, but in this case, it was something that was found around the zygote. Another thing that land plants had to later deal with was the fact that if you look at these charophytes, they're supported in the water, but land plants would have had to deal with a lack of structural support. So if they had a tall body, it would fall over. They also had to deal with a transportation problem. So how are they going to be able to get food and nutrients around their structures? Another problem was that they had to be able to adapt to unfiltered light. So if it's not passing through the water, they're going to get a lot more light, which can be a good thing, but it can also be harmful if you're not ready to deal with that much incoming light. It can cause photooxidation problems. And then they're also going to, if they're able to get out of the water, they're going to have access to more CO2. And more CO2 would enable them to have better photosynthesis. So all of these things are things to think about as we start talking about those tiny little plants inside the shallow waters, the tiny little algae inside the shallow waters, developing into or evolving over time into what we now know as land plants. So land plants were going to have to be able to survive out of water, so they were going to have to deal with dehydration issues, they were going to have support issues, and then they're also going to have some added benefits there where they're going to have some unfiltered light that they'll be able to collect for photosynthesis, and they'll also have more access to carbon dioxide, which should improve their photosynthetic efficiency. So if we look at the kingdom Plantae, this is kind of a general outline of it. You did see a little bit of this in the previous chapter, in the Protist chapter. So it is thought that there was one ancestral algae species that gave rise to the red algae, the green algae, and then what we're calling embryophytes here. So embryophytes is what's typically known as the plant kingdom or kingdom Plantae. Sometimes they group both the charophytes and the embryophytes together, and we call that a single grouping or kingdom. Sometimes the chlorophytes are thrown in there also. In this textbook, we're just going to go with this classification here because that's what your textbook uses. So with the embryophytes, we're talking about plants that have embryos, which certainly does not include the charophytes, and it doesn't include the chlorophytes either. So if we talk about what is unique to the embryophytes, they're going to have a number of derived traits. So derived trait number one is that they did have alternation of generations and then also multicellular dependent embryos. So the alternation of generation part, to refresh your memory, this is when you have multicellular, haploid, and diploid stages. This is something that we did see in the brown algae. We also saw it in the red algae in some of them. This is something that you're going to find in all land plants. If we look at it in this diagram here, we have a zygote. If we start with the zygote is going to be a single cell. That zygote is going to divide by mitosis to produce what we typically think of as the plant. There will be cells within that plant that will undergo meiosis, and then they will develop into spores. Those spores can germinate, and by germinate, they're going to divide by mitosis to produce a haploid multicellular tissue. That multicellular tissue is what we're going to call the gametophyte. So notice that the gametophyte is going to be multicellular, and then down here at the bottom, that typical plant is what we call the sporophyte. It's also multicellular. This is very characteristic of land plants. Now, sometimes one of the stages will be more conspicuous or more obvious than the other one, but they are both going to be multicellular stages. Another thing that is unique or derived in the embryophytes is this idea of multicellular dependent embryos. So we're going to have embryos that actually develop there in the gametophyte. So they will depend on the gametophyte for their water and for their nutritional purposes. It's going to provide them some protection until they're developed enough so that they can then survive without that nutrition from the gametophyte tissue. Derived trait number two is going to be walled spores produced in sporangia. So when we're talking about plants, their spores can actually land somewhere and they can grow. And when they grow, they're going to be dividing by mitosis to produce the gametophyte. So the walled spores here are going to be surrounded by sporopollenin. And sporopollenin we talked about just a little bit ago surrounding the zygotes and the charophytes. So this is the same polymer, but this time it's going to surround the spores. And it's going to provide a resistant outer coat for those spores. Those spores are supposed to be dispersed, and then they have to survive that dispersal process and land somewhere and hopefully grow into that gametophyte tissue. So this is going to help them to do that. Another thing that we're going to have is the sporangia. Sporangia are structures that are designed for spore production. So these are involved in spore production. And within them, within the sporangium, we're going to have cells that are called sporocytes. And those sporocytes themselves actually divide into the spores that we're going to see. So in charophytes, we don't have any sporangia, and we don't have sporopollenin surrounding any spores. If we look at derived trait number 3, this will be the multicellular gametangia. So this is somewhat similar to the first one where we talked about the alternation of generations. In this case, we're talking about those spores. The spores are going to travel somewhere, so they'll be dispersed, and then they're going to undergo mitosis. And after they do that, they will develop into the gametophyte. The gametophyte is going to then produce these structures which are called gametangia. We will have male gametangia, and then we can have female gametangia. The female gametangia is called the archegonia. So this is the female one. And since it's the female one, this one is going to produce the egg. And then the male one is what we call the antheridia. Sometimes these are located on the same gametophyte. Sometimes they're located on separate gametophytes. That would just depend on the individual plant that we're looking at or the species that we're looking at. In any case, this female gametangia, it holds the egg inside, and remember that we're going to have multicellular dependent embryos. So those dependent embryos would develop right there inside the archegonium after fertilization had taken place. The last derived trait that we're going to mention is apical meristems. Apical meristems are places that are specific for producing new cells. It produces new cells by mitosis. And then after these new cells are produced, then they can differentiate into various cell types. Okay. Within land plants, we have the shoot apical meristem, which is the one you're seeing over here. So in this one, you have this area here where you have a whole lot of cell production taking place. And then you have those cells later developing into unique cell types. We also have the same thing taking place in the roots. So this would be a root apical meristem on this side. So it's localized regions of cell division and then later differentiation when we're talking about apical meristems. Those certainly weren't the only derived traits that you find in land plants. There were others. One of them is the fact that plants had to develop a cuticle. That cuticle is going to be a waxy coating that will coat the leaves, it will coat the stem. This is going to help prevent drying out, which was certainly something that land plants would have had to deal with because all of their ancestors were aquatic based. So the cuticle is just one. Another thing that was very important was that they did need to develop a lot of symbiosis relationships with other organisms. We'll see that they did develop a lot of symbiosis relationships with fungi. That helped them to get access to sometimes nutrients, sometimes nitrogen, things that they were not as good at collecting on their own. And then they also developed a lot of what we call secondary metabolites. And secondary metabolites is a broad range of molecules. But by secondary, we're not talking about our typical nucleic acids, amino acids, carbohydrates, those types of things. These are molecules, some of them are for protection, and that could be protection from the environment. So some of them had chemicals that helped them to deal with the amounts of sunlight that they were receiving. We could have ones to help them protect them from, we'll just call it parasites, but we could be talking about herbivores, so little animals that might want to eat them, things of that nature. We have ones that produce flavor for fruit. We have ones that produce a scent, and that might help with seed dispersal. So we have a wide range of secondary metabolites. I just want to point that out. They might have produced a color, but those are going to kind of fulfill all types of roles within that plant. So when you take all of these things together that we've talked about for derived traits, these are things that helped those land plants to do better on dry land. There were benefits to being on dry land, but first they had to be able to cope with the challenges that the dry land would have provided. So just to point out a couple of the major benefits that they would have received from being on dry land, again, number one was lots of sun. So the light is now going to be unfiltered and more accessible, which means that hopefully they'll be able to do more photosynthesis. And then number two is that they also had lots of CO2 or carbon dioxide, which is also going to be good for photosynthesis. So when you take these two things together, this is really going to start to give plants an advantage and help them to really start to diversify once they can just simply survive on dry land.